continue on, and now we'll move on to Stephen Mullin to talk about an update on the Ambroxol trials.
Well, thanks everyone. And I'd like to say thank you for organising this meeting. It's really energising to be in a room with so many people who know so much about GBA, and it really feels like the field's moving forward, particularly the last session. It's great to see so much farmer involvement. My name's Stephen Mullin. I'm an academic neurologist. I now work in Plymouth, but most of my involvement with Ambroxol was when I was working in Tony Shapiro's group in UCL. These are my disclosures. I'm not sure any of them are too relevant for this talk. And I guess I just want to start by saying that the Ambroxol story would not exist without the Gaucher research community. You know, front and centre, they were the ones who brought this forward, and we owe so much to them, you know, for this story, as indeed we do to the whole GBA field. And indeed, you know, this was how it all began. A high throughput screen of potential molecules which would enhance GCA's activity. And one of the top hits was Ambroxol, which was a pH-dependent enhancer of GCA's activity. And subsequent studies that were done indicated that it seemed to bind to the active site of the GCA protein. So most of you will be familiar with Ambroxol, but for those who aren't, this is a drug which you can't actually buy in the UK or in the States, but is available across mainland Europe and other jurisdictions. It's a very old drug, a mucolytic and a cough linctus. Typically, doses are up to about 120 milligrams a day are given, although in the context of respiratory distress syndrome in preterm babies, much higher doses have been given safely without any side effects reported. And it's safe, well-tolerated drug. Patients, particularly at higher doses, will experience a bit of GI upset, and it's reported that there's a higher incidence of kind of anaphylaxis and Stevens-Johnson syndrome, although that's not something I've ever come across. Crucially, it's off-patent, or I should say off-patent for its established indication. The patent situation for a new indication is a bit more complicated, which has been a bit regrettable actually, because I think that really has precluded clinical trials progressing, particularly in the United States, which I think undoubtedly has probably slowed down the field a little bit. But following this discovery of Ambroxol as a potential target, Nir Horowitz's group sort of took the lead and really demonstrated in a dose-dependent manner across a variety of Gaucher genotypes in fibroblasts that it did indeed enhance GCA's activity. And in terms of Gaucher disease, to my knowledge, there hasn't really been a clinical trial which has assessed Ambroxol. Ari gave a very nice talk and overview of the work that's been done so far, and you know, like Ari, I am a believer. I guess the main reason is that enzyme replacement therapy is quite an effective treatment, as is to an extent substrate reduction therapy, and there's not a lot of appetite in, you know, to do an expensive clinical trial on a drug that is off patent. However, this is some really interesting and useful real-world data from China, because Ambroxol has been used off-label in resource-deprived settings where people with Gaucher disease don't have access to these expensive drugs, particularly ERT, which costs hundreds of thousands of pounds a year. And what you can see in this real-world data very nicely is that in patients given it over a long-term period, that there are demonstrable improvements in platelet count, in plasma hemoglobin, demonstrable sustained increases in enzyme activity, and also reductions in glucosylsphingosine sphingosine as well. So whilst there's not a lot of solid clinical trial evidence to support it, I think anecdotally, you know, there is a lot of evidence to suggest that Ambroxol would be a good drug to use in type 1 Gaucher disease. and there's a lot of controversy about Ambroxol, but I think one thing that isn't controversial is that as a small molecular chaperone, it binds to GCAs, which is sequestered, mutant GCAs that's sequestered within the endoplasmic reticulum, causes conformational reformation, and allows passage to the lysosome. It should be noted, as others have pointed out, that Ambroxol binds to the active site of the GCase protein, but then when it gets to the lysosome, it's eluted in a pH-dependent fashion, and so is able to act within the lysosome. Although I think there is still an unknown question as to what the impact of having an active compound that is blocking the active site of GCase outside the lysosome, but I'm not really going to go into that too much today. So it wasn't long before groups such as my old group started becoming interested in this in the context of Parkinson's, and what they were able to demonstrate was that Ambroxol given in cell culture, this one's in fibroblasts, was able to reduce synuclein levels. And this data has been replicated in a variety of models, so in mouse brain lysates and IPS, and interestingly, more recent studies actually suggest there might be a reduction in tau as well. But I guess the thing that was the uncertainty really was in terms of brain penetration. So we knew Ambroxol did penetrate the brain because animal studies showed at very high doses that there was toxicity and neurological symptoms. But when I came into this story, which was around about 2013, we didn't really know what dose to give this at. And once again, the Gaucher research community came to our rescue because this study conducted in Japan, a phase 1 study in a group of 5 neuronopathic Gaucher disease patients, provided really invaluable data that informed the field. So this study did suggest some clinical efficacy, although the numbers are so small that it's difficult to present that. So I'm not going to go into that too much. But what I will say is that it did demonstrate that that Ambroxol penetrated the CSF, and indeed they were able to show within the CSF reductions across every patient in glycosylsphingosine, suggesting that there was target engagement in the CNS. And this concurred with some data that we were collecting at the time through a collaboration with Erwin Bezard in his lab in China on primates, which was looking at brain lysates of primates at different doses to establish what the best dose to use. And what we showed was that at a lower dose, you couldn't detect an increase in GCase activity, but at a higher dose, which corresponds to around about a gram a day of Ambroxol, you were able to see demonstrable increases in GCase activity. And so that really informed the dose choice. For NPD, which was the study which I ran, PI was Tony Shapiro in London. So we called this a 2A, but to be honest, it's really a late phase 1. And this was a proof of principle study. The primary outcome was target engagement. So we were looking to see, first of all, whether Ambroxol got into the CSF, and then to see whether it had an effect on GCase activity. And we were able to show that. So first of all, in all subjects, Ambroxol was detectable, but more importantly, GCase activity changed. It actually decreased. And that may sound a bit counterintuitive, but actually it makes sense. GCase protein floats freely within the CSF. So when Ambroxol binds to it, the activity of the enzyme is likely to reduce. A more unexpected result, which, and to be honest, we nearly missed because this study was conducted on such a small budget, generously funded by Cure Parkinson's, I should say. Sorry, Simon. Was that actually there was a significant increase in GCase protein levels as measured by mass spectrometry. And actually, we very nearly didn't do this assay because it was too expensive. It was only after Kevin Mills in London reduced the price of it that we were able to do it. And I was very glad we did. The other thing that we showed was a small but statistically significant increase in alpha-synuclein within the CSF. So we took that as good evidence to suggest that there was target engagement within the CNS, and so thought this was good enough to move forward to later phase trials. hot on our heels was Steve Pasternak's group. And quite independently of us, actually, Steve had started a phase 2 trial looking at Ambroxol in the context of PD dementia. Now, Steve's here today, actually, and you can talk to him about it, and I'm very grateful that he's shared some unpublished data to show, and he had a poster, and I'm sure he'd be delighted to walk you through it. I think Steve will be the first to admit that this study was probably drastically underpowered to try and achieve what it was looking for. So if you look at Alzheimer's trials in general, they literally have thousands of people in them. And that's because it's very hard to show demonstrable change using cognitive scales. So this study only enrolled 55 participants in the end, of which 8 were GBA positive. And it didn't show a significant change in its primary outcome, which was the ADAS-Cog. Good learning points, though. Well, the first thing is that it was pretty well tolerated. So actually, the only adverse event that was significantly increased compared to the placebo arm was GI upset, which is what you would expect. And this is noteworthy because this was a frailer population with vulnerable brains. So I think this is really, really encouraging. The other thing is that there was some interesting subset data, which Steve was good enough to present to me, which showed that within the GBA positive groups, in those patients receiving high-dose Ambroxol, there did seem to be some change, some improvement in the ADAS-Cog. Small numbers, post-hoc analysis, all of those caveats, but an interesting nonetheless. And then it makes me feel a little bit sad that we now jump to basically 2024, and there hasn't been a lot of completed trials since then, and the trials that are about to complete have only started in the last few years. Roberto Cilia's study in Italy is noteworthy. This is just completed, and I have to say I'm always amazed and very, very impressed by how rapidly the Italians are able to work together to recruit for these studies, because it's no mean feat recruiting 65 GBA positive participants. The primary outcome is a change from normal cognition to MCI or dementia. Lots of interesting secondary outcomes which will broaden our understanding of GBA and Ambroxol's effect on the GCase pathway. I should also mention, and doing this talk was actually a bit of a nightmare because there are so many Ambroxol trials going on at the moment. So I missed a very significant study that's ongoing in Norway at the moment. Similar, although the inclusion criteria are that you have to have established PD-MCI or PDI dementia, and that's due to report next year. So we should have some quite good clinical data in the context of cognitive change in GBA-PD in the next year or so. Likewise, ARI has a trial which is projected to complete in July 2024, so nearly finished. And this is an open-label trial looking mostly at sort of biochemical outcomes, but also tolerance in a cohort of GBA-positive PD patients. There is also a study ongoing in the Netherlands. This is the primary outcome for this is the UPDRS. Interestingly, higher dose selected than most of the other studies. So our study and others selected a dose around about 1.2 grams, whereas this is 1.8 grams. And I'm not quite sure of the rationale for that, because I did try and email the PI, but it was quite late in the day, so she hasn't got back to me yet. But interestingly, that this is due to complete in 2025 as well. And likewise, Simon Lewis is running a platform trial. So for those of you who aren't familiar with it, a platform trial is when you have one placebo arm and multiple treatment arms to try and increase the efficiency of the trial. And one of those two of those arms actually involve Ambroxol. So there's an Ambroxol arm, a doxycycline arm, and then an Ambroxol plus a doxycycline arm. Interesting to note that whilst all of those other studies are exclusively enrolling GBA-positive participants, this one is not going to be doing any genotyping at all. But the outcome is the MDS UPDRS as well. And finally, my old group in London, we'll be running a UK-based multi-centre phase 3 trial that will be opening in Q4 of 2024. This will be enrolling GBA1 positive and GBA1 negative participants. There's a target 50-50 split, and there will be an optional CSF sub-study. And so that will be quite a long and quite a big study, which hopefully should definitively give us an answer of whether this cause improves motor function in PD. So, in summary, and I'm sorry that was all a bit whistle-stop, because as you can see, there is an awful lot of activity in this area. You know, we've established that within the CNS that Ambroxol achieves good target engagement, I think. Its effect on the synuclein pathway is less clear. It's unfortunate that we don't have any placebo-controlled efficacy data to date, and I'll talk about that in a second. But there are a number of phase 2 trials with outcomes, both looking at motor features of Parkinson's and also cognition, which are due to nearly complete. And those also come with an awful lot of PET, MRI and CSF data, which I think will be really helpful to deepening our understanding of how Ambroxol potentially works and how it may impact on these pathways. And as I say, there's a phase 3 trial that's due to open in 2024. And just to end with, you know, a few reflections from me. Well, for anyone who's run a sort of smallish trial, you'll know that you get pretty close to the patients involved, and so it's quite a source of personal regret to me that it's taken so long in between our study completing in 2018 that we actually have started phase 2 studies to try and further this work. And I saw an awful lot of hope invested in this compound by those patients. COVID didn't help. It was very disruptive, I think. The other thing that's been a major problem is reformulation. So this is one of the problems in non-commercial trials. We were having to give our patients 21 tablets of Ambroxol because the established dose was just so much lower than what we had to get to achieve CNS penetrance. And the feedback we got from patients is this isn't going to fly in a larger stage trial, but it takes a long time to get this organised. And I think that's been an issue for a lot of these trials as well. I think an issue that has been problematic and is going to become more and more problematic because of all these commercial trials that are in this space as well, is there are just not enough genotype patients around. Good example, and you know, there are good examples where people have overcome this in Israel and Italy, and that is where really that people across the country have got their act together and have a sustainable and a comprehensive genotyping program early in order to have patients to fuel these studies. And I have to say, I think probably there has been a lack of coordination internationally. There seems to be a lot of phase 2 trials going on, and I think maybe some consolidation of those trials would have meant that we could have been a little bit further along. So I'm really glad to see a meeting like this happening, and hopefully that will, you know, aid that process and mean that we're not going to have to wait another 5 years for more useful results in terms of Ambroxol. So thank you very much. That's all I've got to say.
We've got time for one question. I'll ask a question real quick. Have you looked at, is there any data on effects on GQ substrate levels in CSF following Ambroxol treatment in these studies?
So we weren't able to detect it in the CSF. Oh, excuse me. So in our study, we weren't able to detect it in the CSF, and I think that was a few years ago now. So assays have improved, but yeah, we weren't able to detect it. I don't know of any others at the moment. So, yeah.
Thank you.